It's my pleasure to introduce Dr. Fei Chen for today's seminar. He is a professor of pathology and co-leader of the oncogenic drivers and mechanisms of carcinogenesis program at SUNY Stony Brook, which he has a picture of right here. Dr. Chen's research centers on environmental risk factors and epigenetic regulation in human cancer, a topic of interest to many in this room. He received his medical degree from Nantong Medical College in China in 1982 and a Ph.D. in immunology from Peking University Health Sciences Center in 1994. He did a postdoc at Hershey Medical Center, the College of Medicine of the Pennsylvania State University in Hershey, Pennsylvania, in pathology, and then worked as a genetic fellow and senior service fellow at the National Institute for Occupational Safety and Health in the CDC in West Virginia. In 2010, he was recruited to Wayne State University as an associate professor with tenure in pharmaceutical sciences and was promoted to full professor in 2013. He enjoyed much research success at Wayne State, including being named the Charles H. Gershenson Distinguished Faculty Fellow. He was recruited to Stony Brook in 2021, and he has served on numerous NIH study sections and trained graduate and undergraduate students, as well as postdoctoral fellows. He has 116 publications, and his research is currently funded by an astonishing 3 NIEHS R01 grants. So we're very excited that he'll share some of his research with us today.
Thank you. Yeah, thank you, Caroline, for your warm introduction. And thank you, everyone, for taking your time from your busy schedule to attend this seminar. And first, I would like to thank Dr. Chris State and Dr. John Weiss for inviting me to visit this beautiful university. Actually, I had known Cardinals for many, many years for the basketball team and football teams. But this is my first time to actually visit the campus of this beautiful university. As Caroline introduced, I was recently moved to Stony Brook University from Wayne State University. And this my lab is in the Stony Brook Cancer Center. The building was opened just in 2020. And so the laboratory is pretty new. And this is the Stony Brook Cancer Center. And this cancer center is currently trying to get an NCI designation. And in addition to the clinical part of the cancer center has 5 research programs, including the lipid metabolism in cancer program, the image and the biomarker program, and the oncogenic drivers and mechanism of carcinogenesis program, and the community outreach program and population science program. And my faculty appointment was in the Department of Pathology in this building. Okay. And my laboratory has, can you hear me without the mic? Okay.
I think the people online need to hear.
Oh, okay, yeah, okay. Yeah, my laboratory has a history to study the environmental risk factors and human cancer. And currently research projects in my laboratory including, first is try to understand how the arsenic transformed cells lost its capability to induce the reactive oxygen species. And in this study, we found that arsenic is highly capable of damaging the mitochondria, therefore can affect the reactive oxygen species production. And the second project is about the non-coding RNA in arsenic carcinogenesis. And the third project is we are trying to understand how transcription factor NRF2 can induce the metabolic reprograms during the arsenic-induced generation of the cancer stem-like cells. And the terminology of the cancer stem cells may be not very accurate, just like I discovered the chairman before. This should be the cancer stem-like cancer-like cells, not exactly cancer stem cells. Okay, and the fourth project is we are currently developing, and I will tell you today, is about the epigenetics and the metastasis of the breast cancer, and with an emphasis on the MDIG gene, the mineral dust-induced gene, and the histone 3 lysin 36 trimethylation in the metastasis of breast cancer. In the literature for the MDIG gene means the mineral dust-induced gene. And in the literature, there are several other names, including minor 53 and new 52, and ribosomal hydroxylase 2. Okay. And before I go into detail, I will tell you a brief history about the MDIG gene. And this building, and Caroline is very familiar about this one. This is Hershey Medical Center. And in 1994 to 1998, I was a postdoc fellow in the Department of Pathology at the Hershey Medical Center. Dr. Lawrence Demers was my then supervisor in the Department of Pathology. And he has a research project at that time, was supported by the US Bureau of Mining, and to understand the molecular mechanism of some occupational lung disease, such as the co-works pneumoconiosis, COPD, lung fibrosis, and silicosis, etc. And at that time, we recruited 21 core workers who were working in the mining industry for several years. And also these people had suffered from some chronic lung disease, such as lung fibrosis, COPD, etc. And also we recruited 10 normal control subjects. And the clinician, the pulmonologist helped us to do the lung lavage, to collect the lung lavage cells. And the majority of the cells are in the lavage fluid are the alveolar macrophages. And at that time, my task is to collect to extract the total RNA, then run the differentiation display RT-PCR. So this technique nowadays nobody used it anymore. But at that time in the 1990s, this technique is just like today's the RNA-seq. Try to use this technique to determine the gene expression patterns between the different disease status or condition. And in this differentiation display RT-PCR, we noticed that some RNA species was expressed only in the alveolar macrophages from the core miners, but not from the normal subjects. Okay, and then we did the cloning and amplification and did the DNA sequencing. Also, this is the old fashion of the DNA sequencing. And this is just the antique images of the DNA sequencing. And we used at that time, we used lots of P32, P33 radioactive materials to do the DNA sequencing manually. And when we get the DNA sequencing and search the gene bank at that time, you can see this is handwriting for the DNA sequencing. And in the gene bank, at that time, in 1996, there was no any homolog sequencing in the gene bank. But nowadays, if you put the sequencing into the gene bank, you can see hundreds homolog DNA sequence. But in 1996, now homolog sequence is there. And so therefore, we believe that we identified a new gene from the alveolar macrophages from the core miners. So this gene, because these people are working in the mining industry, exposed to the mineral dust. So we give a name called mineral dust-induced gene, or MDIG. And also this name is exactly reflects the worker characteristic of the mining, just for the mining digger the tunnel. Okay. After several rounds of the amplification and cloning and sequencing, we learned that the gene, MDIG gene, encodes a protein with 465 amino acids. And there is a conserved domain called the Jumonji C domain. And at that time, we have no knowledge about what the Jumonji C domain is. Only thing we know that the Jumonji is a Japanese pronunciation for the cross or cross form. Okay. The Jumonji gene was identified in 1995 by Takashi Tokuchi, and who used the gene trap technology to delete this gene, and found the deletion of this gene caused the defects of the neural plate closure in the embryo mice. okay, creating a horizon glove close to the normal glove. So therefore, they named this gene as the Jumonji gene. And in the literature nowadays, if you put the Jumonji as a keyword for the PubMed search, there are about close to 3,000 papers there. And between 1995 to 2006, only 30 or 40 papers there. And nowadays, we know that many proteins have the Jumonji homolog domain, including the Jumonji gene C-terminal domain and the Jumonji gene N-terminal domain. And this C-terminal domain is usually about 100 amino acids, and the N-terminal domain is 40 to 50 amino acids length. Okay. And also before year 2005, we have no knowledge about the Jumonji C domain. And since year 2006, several groups noticed that the proteins have the Jumonji C domain are most likely the histone demethylases. Okay, so nowadays there are hundreds of histone demethylases from the mammalian cells, from the bacteria, from the fungus, etc. And the majority of the histone demethylases, in addition to the conserved Jumonji C domains, there are several other conserved domains, including the Ph.D. fingers, Tudor domains, and alloid domains, etc. But interestingly, for the MDIG gene, except the Jumonji C domain, no other conserved domain. So this maybe can explain the specificity of the MDIG genes on the several different histone methylation markers. Okay, for majority of other histone demethylases, because of the other domains, so these demethylases are relatively specific for the particular histone methylation marker, such as histone 3 lysine 4, histone 3 lysine 9 trimethylation. But for the MDIG gene, it can target the histone 3 lysine 9 trimethylation, histone 3 lysine 36 trimethylation, 27 trimethylation, and histone 4 20 trimethylation. Okay. And before year 2006, the people believe that the histone methylation is a permanent epigenetic markers, meaning that this methylation is irreversible. Since the identification of the histone demethylase, nowadays we know that the histone methylation demethylation is a dynamic process. Okay, for example, for the lysine residues, this lysine residue can be methylated, catalyzed by the histone methyltransferases. And this methylated lysine residues can also be demethylated by the histone demethylases. So this is a dynamic process. For the lysine demethylation, in general, this demethylation process is a two-step reactions. Okay, in the presence of the Jumonji C family histone demethylases and folate and alpha ketoglutarate and oxygens, the methyl group can be hydroxylated to form hydroxyl methylated lysine residue. That's the first step. So the second step is the hydrolysis of the hydroxyl methyl group to generate formaldehyde. Okay, so the lysine residue is demethylated. Okay, this is the general steps for the histone demethylations. And all of the histone proteins, histone protein H1, H2, H3, H4 can be methylated and demethylated. However, the most extensively studied and the most documented, most important methylation is happened on the histone H3 N terminals. Okay, the lysine 4, lysine 9, 27, 36, 79 can be mono, di, or trimethylated. Okay, and catalyzed by the relatively specific group of the methyltransferases, such as the SMID family for the lysine 4 trimethylation, SUV39 family for the histone 3 lysine 9 trimethylation. And for the PRC2 EZH2 is very important in the cancer development for the lysine 27 trimethylation. Okay, depending on which lysine residue is methylated, the gene transcription outcome or gene transcription dynamic will be different. For example, the lysine 4 trimethylation was considered as an active transcription marker through the formation of the euchromatin. And in the euchromatin region, the genes can be actively transcribed. Okay, so therefore the lysine 4, lysine 79 di or trimethylation can facilitate the gene transcription. So from the epigenetic point of view, so this methylation called active epigenetic markers. Okay. In contrast, the trimethylation on the lysine 9 and the lysine 27 of histone 3 was a replacement histone methylation or epigenetic markers due to the formation of the heterochromatins because the nucleus are compacted together. And in the heterochromatin regions, the genes are inaccessible by the transcription factor. So therefore, this trimethylation can replace the gene transcription, cause the gene silencing. And the first evidence of the MDIG gene is the histone demethylase is from our lung cancer study. And at that time, we screened 25 human lung cancers, lung tumors. Each was a case matched with the normal lung tissues. And we found an inverse relationship between MDIG expression and histone 3 lysine 9 trimethylation. Okay, except this large cell lung cancer, all other lung cancers showed the high level of the MDIG expression, but the histone 3 lysine 9 trimethylation was decreased. Okay, and also I didn't show you here. And in the in vitro test tube reaction, we used the recombinant, both the recombinant MDIG proteins and the immunoprecipitated purified MDIG proteins, and we can see some histone demethylation activity in the test tube reactions. Okay, and then we also checked the expression profile of MDIG in several different types of the cancers. And in the lung cancer, because the original focus of our study is human lung cancer, and we believe that MDIG is the oncogene for the lung cancer development. And we screened 250 human lung cancer samples. Each one has a case matched normal lung tissues. And about 70% of the human lung cancers showed a high expression of the MDIG gene. This is just use the RT-PCR reaction. Okay, and also we checked 25 hepatocellular carcinoma. Okay, and about 70 to 80% of the hepatocellular carcinoma showed the high expression of the MDIG genes relative to the normal hepatic liver tissues. Okay, for the breast cancer, at that time, we only collected 12 human breast cancer samples without the normal control breast tissues. And in this screen, we only found 4 samples expressed the MDIG, and 2/3 of the samples without the MDIG expression. Okay, indicated to us that in the breast cancer, the percentage of the MDIG expression is very low. Okay. So then we used the online available database to check the MDIG expression in the different stages and the different histological types of the breast cancer. And in general, we found that the early stage of breast cancer, stage 1, stage 2, express a high level of MDIG than the later stage of the breast cancer, the stage 3 and stage 4. Okay, for the different histological subtypes, we found that the medullary breast cancer and ductal carcinoma in situ and methanous breast cancer express a high level of the MDIG. In contrast, for the invasive ductal carcinoma and the invasive lobular carcinoma, the MDIG expression is lost. Okay. And just recently, this is just a few months ago, we also checked the hormone status and the MDIG expression. And for the hormone positive breast cancer, we found that about 50% of the breast cancer samples showed the MDIG expression, and another 50% is MDIG negative. But for the triple negative breast cancer, the positivity rate for the MDIG is much, much lower than the hormone positive breast cancer. Only about 15 to 18% samples showed the MDIG positive. Okay, more than 80% of the triple negative breast cancers are MDIG negative. Okay. And so this is another publicly available database search for the gene expression and histology or pathology of human breast cancer. And we've noticed that for the metastatic breast cancers, the MDIG expression is lost. Okay, so this data is correlated with the patient survival data. Okay, in general, the breast cancer without lymph node metastasis or the total breast cancer, okay, the high level of MDG expression predicts the poorer survival for the overall survival for the breast cancer patients. However, for the breast cancer sample with the lymph node metastasis or the triple negative breast cancer, the high expression of MDG expression is beneficial for the patient survival. So this data suggests to us that for the early stage of breast cancer, without the metastasis, the MDG is very likely an oncogene. It can promote the cancer development. But for the later stage breast cancer or metastatic breast cancer, this cancer lost the expression of the MDG. And if the MDG expression level is high in this metastatic or triple negative breast cancer, so this MDG may be functioning as a tumor suppressor that can improve the patient survival. Okay. And to understand how MDG contributes to the pathogenesis of the breast cancer, and we use the CRISPR-Cas9 gene editing technique to knock out the MDG in the triple negative breast cancer cells, MDA-MB231 cells, and T47D cells, and MCF7 cells. And in here, I just show you the MDA-MB231 cells. Okay, and we screened the clones subject to this gene editing, and this wild type clones also subject to the CRISPR-Cas9 gene editing, but failed to the knockout of the MDG. So therefore, we picked up this clones as the wild type clones, and also we picked up the clones have complete deletion of the MDG gene, and then we used the wild type cells and the knockout cells and inoculate these cells into the NSG immunodeficient mice into the mammalian fat pad. Okay, we used the 5 mice for each group, and here just show you 3 tumors from each group. And this is the wild type cells, and wild after 7 weeks, the wild cells can form the large size of the tumor. Okay, but for the knockout cells, the tumor size is only about 1/3 of the wild type cells. So this is confirmed our hypothesis. The MDG is the oncogene. Okay, it can promote the tumor growth. If you delete MDG, without the MDG, the tumor growth will be delayed. So therefore, generate a small size of the tumor. Okay. However, the story is more complicated. When we checked the metastasis of the mice in the mice, and we found this lymph nodes from the wild type mice. This is the axillary lymph nodes and the lumbar lymph nodes, the peritoneal lymph nodes. Okay, this is the number and the size of a lymph node from the normal mice. But for the mice received the knockout MDG knockout cells, remember this tumor size is much smaller than the wild type cells. But for the metastasis, we can see that both the number and the size of the lymph nodes, the knockout cells generated much larger in this mice. Okay, and also for the liver metastasis, okay, the knockout cells is more active for the liver metastasis, okay, than the wild type cells. Okay. So this showed us that the MDG somehow can inhibit the metastasis. And also this was confirmed by in vitro cell motility, invasive migration assay. And this is wild type cells, this is knockout cells. Okay, and in general, the knockout cells is more active in invasive migration in the in vitro cell culture. Okay, so therefore, we can conclude that MDG can somehow inhibit the cell motility or cancer cell metastasis. Okay, to further understand how MDG affects the cell metastasis or cell motility, we did the quantitative proteomics study. Okay, and this just shows you the top 25 most differentiated expressed proteins between the knockout cells and the wild type cells. This is the most top 25 downregulated protein in the knockout cells. And this is the top 25 most upregulated proteins in the knockout cells, more than 5-fold increase relative to the wild type cells, the proteins. And interestingly, among these top 25 proteins, 9 of these proteins whose genes are in X chromosome. So this is totally new to us. And I checked the literature, and I didn't find many information regarding the relationship of the X chromosome and the breast cancer metastasis. Okay, so this may be indicating to us that the X chromosome, the genes in X chromosomes, maybe play some important roles for the breast cancer metastasis, such as the genes for the MAGED2, I will tell you a little bit later, and several other genes in the X chromosome. Okay, and we did the functional pathway assay for the upregulated protein in the knockout cells. And in the top ranked pathways of these proteins are involved in the cell motility, such as the catalytic binding, actin binding, G protein bindings. Okay, so indicating to us that it is true. Knockout MDIG can increase the expression of the genes contributing to the cell motility or metastasis. This was not surprising for the downregulated gene because we already know that MDIG can affect the ribosomal RNA transcription and the ribosomal biosynthesis. So therefore, knockout MDIG can downregulate the genes or protein in this pathway for the ribosomal biosynthesis. Okay. And to confirm this proteomics data, so we also did the Western blot to check the protein expression in 5 wild type clones and 12 knockout clones. Okay, and we found that the knockout MDIG can decrease the expression of the oncogene c-MYC, and to a least degree is the EGF receptor alpha. Okay, knockout MDIG can downregulate the expression of this oncogenic genes. For the EMT marker, epithelial mesenchyma transition markers, for the E-cadherin-1, the data is not conclusive because we see that both the wild type and knockout clones, some knockout clones, express this epithelial marker E-cadherin. And for the mesenchyma marker, SNEL, vermetin-1, and twist-1, and the wild type cells express high level of SNEL1. In contrast, for the other 2 mesenchyma marker, Wetman-1 and twist-1 was highly expressed in the knockout clones, but not in the wild type clones. And also these 3 proteins, the MAGD2, Xepsin D, lysosome proteins, okay, and a cytoskeletal protein, stathmin-1. In proteomics analysis, we found these are among the top 25 most upregulated genes, and it is true. In the Western blot, we confirmed that the MDIG knockout can increase the expression of these proteins. And at the beginning, I said that MDIG maybe a histone demethylase can affect the methylation of the histone demethylation. So therefore, the next question we would like to ask: does the histone demethylase activity has something to do with this effect of MDIG on the cell motility or the protein expression related to the cell motility and the cancer cell metastasis? Okay, we did the CIEHS analysis, and in general, we found that the knockout clones, the level of the histone 3 lysin 36 trimethylation is much more enhanced or enriched in the genomes. Okay, this just show you 2 examples in the X chromosome and the chromosome 9. Okay, in general, the enrichment peak in the CIEHS for the histone 3 lysin 36 trimethylation in the knockout cells is much more than the peaks in the wild type clones. Okay, suggesting to us that the MDIG is truly involved in the demethylation of the histone 3 lysin 36 trimethylation. Okay, if we knock out MDIG, the histone 3 lysin 36 trimethylation will be enhanced. Okay, in other words, in the normal level of the MDIG, it can suppress the histone 3 lysin 36 trimethylation. Okay, we also checked the genes, which showed the more than 2-fold increased enrichment of the histone 3 lysin 36 trimethylation in the knockout cells, and for the functional pathway assay. And the top ranked pathways are in the integrin interaction and the base excision repair. Maybe this one, Chris, maybe you may be interested for the base excision repair. Okay, and also for the G protein interaction, etc. Again, suggestion to us that the histone 3 lysin 36 trimethylation are very likely regulating the expression of the genes involved in the cell motility or cell metastasis through the interaction with the beta integrin or G proteins. Here, just to show you the histone methylation profiles for the selected top 25 most upregulated protein in the MDG knockout cells. Okay, all of these genes are listed here. You can see the significantly enhanced enrichment of the histone 3 lysin 36 trimethylation in the knockout cells than the wild type cells. Okay, and also we checked other methylation marker, the lysin 4 histone 3 lysin 4 trimethylation and active transcription marker. And for the repressive marker, histone 3 lysin 9 trimethylation and histone 4 lysin 20 trimethylation. Okay, the most effective, most significant difference is the histone 3 lysin 36 trimethylation. Okay, for the other change of the methylation, the change is very marginal. Okay. To determine which gene that was upregulated in the MDG knockout cells are playing significant roles in the cell motility or cancer cell metastasis. Okay, so we picked it up. We were very scrambled at the beginning because we have no knowledge or idea which gene is the most important in mediating the increased metastasis of the MDG knockout cells. So we just randomly, it's totally randomly, picked up the gene, the MAGD2 genes, and in the ChIP-seq, the MAGD2 gene showed increased the histone 3 lysin 9 trimethylation in the gene body of the knockout cells. Okay, and in the database, in the publicly available database, and it showed that the breast cancer tumors express a high level of the MAGD2, and the highest expression level of the MAGD2 is in the metastasis, the metastatic breast cancer. Okay, and also there is a negative correlation between MDG gene and the MAGD2 expression in 7,500 human breast cancer samples. Okay, and for the patient survival, for the overall survival of the triple negative breast cancer, okay, the high level of MDG, MAGD2 expression can predict the poorer patient survival of the breast cancer patients. Okay, so this suggested to us that MAGD2 may be an oncogenic factors can contribute or plays critical roles for the metastasis of breast cancer. And so to partially confirm this, we use the siRNA to silence MAGD2 in 3 MDG knockout clones. Okay. After the silence of the MAGD2, then we checked the in vitro invasive migration capability of the cells with the control siRNA or MAGD2 siRNA. Okay, and you can see the silence of MAGD2 can reduce the capability of the invasive migration of the cells in vitro cell culture. Okay, so our next experiment is trying to use the mouse model to knock out MAGD2 to see whether or not MAGD2 is truly important to affect the metastasis of the breast cancers in vivo. Okay. For the MAGD2 genes, actually the information right now is very limited in the literature. The only thing we know that the MAGD2 gene is in X chromosome, okay, located in the X-linked intellectual disability region of the X chromosome. And normally the protein is located in the nucleoli, the cells. And we know that what we learned that the genetic mutation or polymorphism can affect the expression of the sodium, potassium, and chloride co-transporters in the kidney tubules. Okay, because of the decreased expression of these co-transporters, it can cause the disease called the antenatal Bartter syndromes, featured with polyhydramnios and salt loss and polyuria and hypokalemia. Okay, this is for the newborn baby. Okay, and also we know that the MAGD2 can interact with some E3 ubiquitin ligases and heat shock protein and G proteins in the ER. Okay, and then therefore the MAGD2 maybe can affect the protein maturation or packaging after the translation. Okay, in the ER. Okay. Okay, this is what I would like to share with you today, and I would like to summarize what I said so far. Okay, in the breast cancer, in the early stage of breast cancer, the MDIG expression level is high, and in the late stage and the metastatic breast cancer, MDIG expression is lost. Okay, MDIG is a histone demethylase. It can antagonize the histone methylation, histone 3 license 9 trimethylation, histone 3 license 27 trimethylation. Okay, because these histone markers are replacement markers for several oncogenes, such as c-MYC, EGF receptor, and some ribosomal RNA. Okay, due to the MDIG repression on these replacement markers, okay, therefore the oncogenes was activated or actively expressed. So therefore, in the early stage, when MDIG is high, so MDIG maybe they promote the tumor growth. Okay, and in the later stage of breast cancer or metastatic breast cancer, due to the loss of the MDIG, okay, and MDIG is also antagonistic for the histone 3 lysis 36 trimethylation. Okay, due to the loss of this inhibition of the MDIG on histone 3 lysis 36 trimethylation, so therefore the genes involved in the cell motility or metastasis. Here, just to give you examples such as the MAGD2 I showed you before, and another gene called IQ-SEC2. Okay, and these genes are regulated by the histone 3 lysis 36 trimethylation. Okay, therefore, in the later stage of breast cancer or metastatic breast cancer, the tumor metastasis is predominant. Okay. Finally, I would like to thank the people who made contributions to this project. The people from Penn State University in 1990s when I was a postdoc fellow in Hershey Medical Center, and the people from Wayne State University, Stony Brook universities, and my two collaborators from German Cancer Research Center at Cologne University in Japan. Okay, and also I would like to thank the funding agency, NIH and the CDC for their support to this project, and also the support from Stony Brook University and Wayne State University. Okay, I would like to entertain questions from you. Thank you.
This might be sort of a naive question. Interested in MDIG and X inactivation.
Yeah. That's an excellent question. And yeah, for the normal cells, we know that the only one X chromosome is active, another X chromosome is inactive. But in the cancer cells, we don't know the status of another X chromosome. Maybe the inactive X chromosome, such as the what's called bar body, right? The inactive X chromosome, yeah, maybe reactivated in the cancer cells. There's one possibility. Maybe MDIG in the normal level, MDIG can contribute to the inactivation of one of the X chromosome. But without MDIG or deletion of the MDIG can facilitate the reactivation of the inactive X chromosome. This is one possibility. Another possibility is that the MDIG, maybe only, no matter it's normal cells or cancer cells, still only one active X chromosome. That's a hypothesis. The scenario is another X chromosome is still inactive. Okay, but MDIG can somehow increase the expression of the genes from the active X chromosome. So this is my hypothesis. But certainly we need to do more studies to see whether or not the X chromosome status in the cancer cells, especially in the triple negative breast cancers, are changed in response to the MDIG expression levels or other genes. Okay, yeah, this is a good idea. Maybe we need to do something such as for the inactivation of the X. I think some genetic method can determine whether or not the inactive X chromosome can be reactivated or not. And also a very simple pathway, maybe we can use the immunohistochemistry to use the X chromosome marker. So actually one of the epigenetic markers, the histone 3 lysin 27 trimethylation, is highly enriched in the inactive X chromosome. Maybe we can use this immunohistochemistry, fluorescent microscope to see the X chromosome status in the breast cancer cells.
I think there's needs to, like, I know what the CIEHS.
Oh yeah. No, we didn't do this. Yeah, maybe we can. Yeah, that's a good idea. Yeah, to check the XIST level of the in the wild type and the knockout cells. Yeah. Hi.
Question on the lung cancer and one question on the breast cancer.
Okay.
I didn't show all the data from the lung cancer, but you originally identified MDIG from the alveolar macrophages. So, in the rest of all the things that you showed, how do you know it's regulated the same in the actual cancer cell in the lung cancer thing? It's not just a readout for alveolar macrophages, especially in pollution-induced.
Yes. Actually, this is an excellent question. Even for the originally, we identified this gene from the lung lavage cells. Actually, in the lung lavage cells, this is a mixture of the different cell types. Although I said the majority of the cells are alveolar macrophages, but actually at that time we didn't distinguish clearly. It is truly from the alveolar macrophages, or from epithelial cells, or from fibroblasts. But certainly it's not from the neutrophils, because in the lavage cells, we just culture the cell lavage cells for 24 hours. We only collect the adhesive cells, and suppose the majority of the adhesive cells are the alveolar macrophages. And for the cancer, for the lung cancer, and we know that 90% or more than that, 90% of the lung cancers are derived from epithelial cells. Okay. And from the different cell type, actually, we did the checked the explained pattern of the MDIG among the different types of the lung cells, including the alveolar macrophages, bronchoepithelial cells, lung fibroblast cells. And we found this is under the disease condition, not the normal condition. Under the disease conditions, all of these cells are able to express the MDIG. Okay.
In the breast cancer, so do you know that the phenotype driven by knockout of MDIG is actually due to loss of the demethylation activity? So if you re-express MDIG that was demethylation deficient versus wild type, can you rescue those phenotypes?
Yes, actually, looks like you read my grant proposal. Yes, actually, this is one of this is we are developing a new NIH applications, and this is one of the M. And we try to actually, this is the in vivo data is very important. It's critical to confirm whether or not the MDIG is truly important for the metastasis. Because right now we just centered on the cell culture model or cellular model. And certainly even in the cellular model, just like you said, we need to re-express the MDIG into the knockout cells to see whether or not the cell motility or the metastasis in the mice can be changed, okay, can be reversed. Yeah, this is a very important aspect in our next plan we will to do. Yeah, and in addition to the MDIG, and also we are planning to check the MAG D2 I mentioned you earlier. Because in the literature, there is no detailed information about this gene in the breast cancer metastasis. And we also use both the knockout and the rescue experiment to see whether or not this gene can truly affect the metastasis of breast cancer in vivo. Yeah. Thank you for this question. Okay. Yeah. Hi, Karen.
You looked at, speaking of metastasis, I have a couple questions. One, in your MDA-MB1 model, was implanted in the real fat had an actual metastasis?
Yeah, this is in the mammary fat pad. Yeah. Yeah. This is not a subcutaneous, but the mammary fat pad. Yeah. Mhm. Mhm. Yeah. And the metastatic tumors. Yeah. Actually, this is we had a, we are trying to get these human samples, and at this moment, because I contacted the clinicians in the Stony Brook Cancer Center, and there is a biobank, and they collect some human breast cancer samples, but unfortunately, the information I learned that from the Stony Brook Cancer Center, they said they didn't have the metastatic breast cancer samples. They only can get the primary breast cancer samples. Because this is in general, because for the breast cancer patient, if they already metastasized, and the patients are reluctant to do the second surgery to get the metastatic sample. Yeah, I will. Yeah, I will contact the Sloan Kettering Cancer Center. Yeah, to this. Yeah, to look at it. Yeah. Thank you for sharing this information. Yeah. Hi, Mike. Oh, thank you.
So I was wondering, do you have any idea what causes low expression?
Yeah. That's the terrific questions. There are several possibilities, and one of the important possibilities is during the cancer progression, maybe some additional epigenetic patterns changed. One of the epigenetic changes is the DNA methylation, and I know many people are interested in the DNA methylation. And we checked the publicly available database for about the DNA methylation, and to see whether or not the different stages of the breast cancer showed the different pattern of the DNA methylation, and also on the DNA methylation for the MDIG gene, particularly. And for the MDIG gene, the data is not conclusive, but in general, the difference is very marginal for the DNA methylation, and it looks like in the later stage of the breast cancer, the total DNA methylation level is decreased, but it's not very significant, and some data is contradictory. And so, but we still think that this may be the different region of the DNA. For example, if we focus on the MDG gene, either the 5 terminal of the 5 terminal genes, or the 3 terminal of the genes, or the gene body of the DNA methylation may be changed at the different stage of the breast cancer. And so this methylation can affect the expression level of the MDG. Okay, this is one possibility. Yeah, maybe certainly there are other possibilities.
Yeah, I was thinking because you said it interacts with E3, so I was wondering whether it's one.
Yeah, it's possible, highly possible. Yeah, we will test this. Yeah. Thank you. Yeah. Hi, Caroline.
Non-coding. Are there any proximity? We did the RNA-seq study for this one, for the wild type and knockout cells.
which included the non-coding RNAs also. And the data was not summarized yet, but we found that a special group of the non-coding RNAs can be affected by the, I didn't remember which pathway, but somehow can be affected by the MDIG expression status. Yeah, and also, yeah, one piece of data I showed here is that the knockout MDIG can downregulate the expression of the ribosomal RNA or the small subunit RNAs. So it is highly possible that MD can affect the other linker RNA expressions. And so therefore it can change the overall landscape of the epigenetics in the cells. And we know that the non-coding RNA, including the microRNAs and some other long size of the RNAs, are very important to affect the gene expressions and the chromatin configurations, etc. Yeah.
We have a few questions online. These are from Dr. Hong.
Mm.
It seems that MDIG knockout has an effect on expression of a selected set of genes via histone methylation, but where does the selectivity come from? How does MDIG after histone methylation and selected genes but not globally?
Yeah. That's a very interesting question, because in our mind, we know that the histone methylation on the genome is global. The histone methylation should be everywhere. Okay, but why, for example, in here, why we found that only a specific groups of genes are affected by MDIG knockout, and by the effect of the effects through the effect in the levels of the histone 3 lysis 36 trimethylation. Yeah, this is the question actually haunted us for a long time. So we believe that in addition to this particular histone methylation markers, maybe the gene structure or other transcription factors or chromatin binding proteins can affect the MDIG or its effect on the histone 3 lysis 36 trimethylation. Okay, in other words, some genes can be regulated by some other chromatin binding proteins, no matter it's the transcription factors or other chromatin modification proteins. Okay, and in addition to the histone 3 lysis 36 trimethylation, or through the interaction of the histone 3 methylation and other chromatin binding proteins, so therefore it can affect the particular group of the genes, such as the cell motility or affect the cell migration invasion itself. So yeah, this is a very difficult question to address, but at least we, in my mind right now, I think there are maybe certain other interactive proteins can determine the relative specificity of the MDIG and histone 3 lysis 36 trimethylation on this particular group of the gene expression.
And then second one from Dr. Holmes, have you tried to overexpress MDIG in normal epithelial cells or breast cancer cells? If so, what were the outcomes?
Yeah, this is just like the earlier question asked that. In the normal epithelial cells, and despite the expression level of MDIG is very low, but the gene is still there. But we didn't do the overexpression in the normal cells. In the cancer cells, for example, in the lung cancer A549 cells, we did overexpress the MDIG. And so in addition to the endogenous MDIG protein, it also expresses the exogenous MDIG proteins. And we try to see we didn't check the histone methylation and the demethylation, but we tried to test the expression of some oncogenes. And few, not all, not many, few of the oncogenes, we can see the overexpression or increase the activity of the MDIG can increase the expression of some oncogenes. Yeah. So our hypothesis, this may be partially support our hypothesis that because MDIG is a histone demethylase, if we overexpress the MDIG, it can suppress the replaceable histone methylation. So therefore can increase the oncogene expression. But in the breast cancer samples, and we will do the rescue expand in the knockout cells because it will give us more conclusive results. Yeah. Yeah. Hi.
Hi, great talk.
Oh, thank you.
Kind of the double-edged sword of high expression increases tumor growth, and low expression increases metastasis. Does it seem like there's a targetable option in terms of therapeutic development?
Yeah, I can. Yeah, actually, from the as a biologist, it is very general. We try to simplify the questions as much as possible, and even the hypothesis I discussed here is extremely simplified model, and in the realities, and maybe the situation is much more complicated. So therefore, for the molecular targeting, from the therapeutic point of view for the breast cancer, if we say that if it is true, the MDIG is important for the tumor growth in the early stage of breast cancer, we can think about the preventive strategy or some etiological factors for the breast cancer development. If we can prevent the MDIG expression, it can prevent the initiation of the breast cancer. Okay, so this is what we can think about it. For the later stage breast cancer, and if MDIG is a tumor suppressor for the metastasis, it is hard to target this one because it is very difficult to overexpress or reactivate the MDIG in the metastasis breast cancers. Practically, I think, yeah. In the experiment, you can do this, but in the clinical, it is very hard because, yeah, actually there are some drug companies are developing the epigenetic drugs to target in the DNA methylation and histone methylation to see whether or not these epigenetic drugs can be therapeutically beneficial for the cancer patients. But so far, I don't think the data are conclusive. Yeah.
That's it. Thank you.
Oh, thank you, Chris. Yeah, thank you, everyone.